So we are continuing with organization of chromatin, organization of nucleus. I see we had some music this morning. I see some art will be coming, and I hope some more of positive topology because that was the term introduced by Jay. So good topic. Positive topology. Yes.
So let me start by saying it's a pleasure to be here at this, I think, very interesting and timely meeting on these topics. I will continue the discussion we started this morning, and I will talk about the organization in space of chromosomes in the nucleus of higher eukaryotes. Because as we have heard this morning, and you know already, chromosomes have a complex organization which displays patterns, and those patterns are also changed. And one of the key questions, as we've discussed, is to try to understand what are the principles underlying such an organization, such a complex organization. And to do that, I will consider very schematic models on about some of the molecular mechanism which could be driving the spontaneous organization of the system. And this will be done going back to classical polymer physics. This is a work done in collaboration with Anna Pombo's lab at MRC Imperial College and Josie Dosti at McGill, and then my group, which now is in Naples. A picture of the kind of ideas and we have in mind has been already discussed this morning by Rez, by Leonid, and the idea is this, that there is chromatin is organized at many different levels. For instance, at levels of single genes. We know in higher eukaryotes, for instance, that a gene can be regulated by very distal non-coding regions, which can be far away from the gene itself, 10, 50 times the length of the gene itself. And to regulate a given gene, those regulatory regions have to fold onto the promoter of the gene and assemble what is called the expression unit for that gene. But intriguingly, you also have that some of those distal regions can control a different gene. So you see you have patterns which change. And the same distal region can control more than one gene. And so there is another problem, not only how you find your target, but how you make the choice, how you change the patterns of organization. And to be short, such an organization is found at a number of levels. One other element is of aggregation is the transcription factor. This is more still debated, but the idea is that there are molecular complexes where you have more than one pole 2. And so those become focal points for co-regulated and co-transcribed genes. And so this is another level of organization in space of co-regulated genes. And transcription factors are only a very sophisticated molecular machine which holds together the genome. We know tons of molecules of specific transcription factors of general chromatin organizers, CTCF, SAT1, many, that have a role in shaping the architecture of chromatin. And as we've listened this morning, the organization goes up to the scale of the entire nucleus in higher eukaryotes. This is a picture of a human cell line, of the nucleus of a human cell. And the different colors are the chromosome territories. So chromosomes are not randomly mixed, but they tend to occupy specific regions. And you see there is a sort of geographical map where each chromosome has preferential neighbors. And those maps are changed in different tissues during differentiation and so on. So once more, a complex organization. And the feeling is that you have an organization which goes through a very broad range of scales. And so this morning we have seen this idea of a sort of scale-free organization, which has been substantiated by the high-C discoveries that we have listened to this morning. For instance, the contact probability, to be precise, the average contact probability of two loci having a given genomic distance. So the idea is this: you take two loci which have a genomic distance given, and you measure the counter probability of those, and you average genome-wide. And you get, as we have seen this morning, sort of power law behavior. So very exciting, scale-free arrangement of chromatin, at least in a domain which goes between 0.5 and 7. And the other exciting news was the exponent, as we've seen this morning, because 1.081 is not found in the counter probability of standard polymer models. And so the idea of the fractal globule model has been introduced, as we know. And the exciting fact is that I call alpha the exponent of the power law, is that the fractal globule predicts an exponent, is a transient state which predicts an exponent which is very close to what you observe experimentally. And you also predict, for instance, as we've seen, power laws for the mean squared distance between two loci. This was also very exciting. And of course the question is, can we describe the variety of behaviors of chromatin with a single state, fractal globule or whatever? So what we try to do is to try to collect together recent data to try to start addressing this question. And I show you some of these results. This is the average concrete probability as a function of genomic distance for a number of recent experiments in human cell lines, which have piled up really in recent months. And the original data by Retz and Job Decker's group is the red one. And you see that in the domain they mentioned, indeed you find a very good description in terms of a power law with an exponent, which is the one they mentioned, 1.08 roughly. But as you see clearly from this picture, that's not universal. If we take the data face value and you consider other systems, for instance, data in blue from Calor et al., you see that if you want to fit that in the same range with a power law, you still can. But the exponent is different, it's 1.3. And if you take the data from Dixon, this is a human stem cell line, you see that this is the magenta one. You can still try to fit with a power law, but the exponent is much bigger, 1.65. And if you go to consider other organisms, Drosophila was mentioned today. This is the data by Sexton et al. In Drosophila, for instance, the exponent is roughly 0.85. And if you get picky and you want to look only at repressed regions in Drosophila, it goes down to 0.7. So different systems can have different exponents. Alpha is not a universal state. And of course, the following question is, but what happens in a given system? And again, this is a collection of data. This is the original data by Retz et al. And again, the genome-wide average is in red. The other colors illustrate the behavior of different chromosomes in that system. And you see there are a number of chromosomes which are really close to the average, by definition of average in a sense. But there are clear departures. Look at the yellow data. This is chromosome X, which is known to be especially compact. And you see this is above the average. And if you want to fit that naively, your exponent becomes 0.9. And another chromosome, chromosome 19, for instance, which is instead genome reach open, it's on the opposite side. It's an exponent which is roughly 1.3. And of course, if you have only one experiment, it's hard to say if these are fluctuations, noise effect, whatever. But you see, also in the different systems, in some of the different systems I mentioned, as those, you have different average behavior, but consistent departure from average of chromosome X and 19, for instance. And so again, the message is, also in a given system, different chromosomes can have different exponents. Once more, there's not a universal exponent per cell. In fact, this is, I think, that the different behaviors of different exponents is not something new. If you go back to fish data, which is this previous technology, less powerful, but you can make very, very good measure with that too. it is known that different loci, different chromosomes have different behaviors. There's plenty of data on that. I show you only 2 examples. And this is the fibroblast chromosome 11 in a recent paper, and this is pro-B cells chromosome 12 in another recent paper. And you see, first of all, you may have a power law behavior, but only in a region of the genome, small scales typically. But, and you see that you usually see a plateau in many of those examples. And second, details matter. Look where, for instance, the plateau sits in here. You see it's roughly 10 megabases. Here, well, order of magnitude, it is 0.1 megabases. So different loci have different behaviors, and we have to take this into account. So we are faced with the fact that there is a variety and the complexity of observed data showing different behaviors of different regions, and there's not one exponent of the counter probability. There is a range of variables. How do we explain that? Well, our starting point was a sort of known idea, an even obvious consideration. The fact that, well, it's the chromatin organization, chromatin conformation, must be shaped by the interaction of chromatin. And chromatin interacts with nuclear envelope, with nuclear bodies, and of course with transcription factors, with molecules, as I've already said. And so by now, let's focus on this system where we only consider the interactions between diffusing polymers, diffusing chromosomes in this pictorial view, and diffusing factors, diffusing molecules. What we want to try to understand in that context is, well, first, how can be different conformation and different scaling properties obtained? Second, how you reliably control architectural changes which are key to the functional behavior of the system. And third, how such a scenario compares with the high sea and fish data I illustrated. And to try to discuss this, we considered an incredibly simplified model, which is the one you see here. We call this the strings and binders. And this is a standard model of polymer physics, where the string is a self-avoiding walk chain, and along the self-avoiding walk, what you have are binding sites for diffusing factors. And that's it. So this stupid model, which of course is a dramatic simplification with respect to what you have in real nuclei. In real nuclei, you have different kind of binding sites, different kind of binding factors. You have crowding effects, which are important, of course. But in this system, this is simple enough to be fully explored, as you know, in polymer physics. It is characterized, this model is characterized by 2 main control parameters. One is the concentration Cm of binding molecules, and that, because if you have no binding molecules, this is just the self-avoiding walk. And the other is the binding energy of the molecules to the binding sites along the string. That's the string and binders model. And this is simple enough to be fully investigated with standard tools of polymer physics. And you can see what are the emergent properties of such a schematic simplified system. Since the audience is mixed, may be worth to give an idea, at least an insight of what are the emergent features, the concept behind this kind of models. I don't want to delve into details of physics, but let me, with a cartoon, illustrate what happens here. And in my cartoon, we consider a polymer where you have 2 binding regions, the red and the blue. And you have binding molecules for those. Consider first, this is the top part of my slide, consider first the case where you have a low concentration of binders. Think about it, it's easy to understand that you will never fold the polymer in a stable way, because to form a stable bridge, you need a sequence of 2 random events. First, floating molecules must find the binding site, say, on the red sequence, in the red region. And then the other region, the blue, randomly floating, must find the same location before the molecules detaches. So you don't need heavy computer simulation to understand that, on average, that polymer is open, although you have the ingredients to fold it. Now consider the opposite limit, where you are in the high concentration regime of molecules. The formation of a bridge is more or less working as I just illustrated. It's easier because you have many more molecules. But the real difference is the fact that when you form the first bridge between the two regions, it's much easier to form a second bridge because the two regions are nearby, and locally you have a high concentration of binders. So you immediately form the second bridge, and this is reinforcing the first and helps the formation of a third. You understand? You switch to a conformation where the two remote regions, the red and the blue, are stably held together, and your polymer conformation now is stably looped. And this argument also predicts the existence of a threshold concentration. In my cartoon, this is the point where the positive feedback mechanism I illustrated becomes winning. In fact, this is the theta point of the polymer, as most of the students knows, and corresponds to a thermodynamic phase transition into the system. This is the phase diagram that you get with this strings and binders stupid model. This is the phase diagram in the space of the binder concentration and of the affinity, binding affinity, as I told you. And you see at low concentration of binders, low energy, there is no way you fold it. You have the ingredients to fold, but your conformation will be spontaneously the one of the open polymer. In the higher region, you have the other phase where the polymer spontaneously folds into a compact lump. And there is the transition line, which is the polymer theta point, where you have the theta point fractal state, a different state. Notice the ranges on the two axes. Look first at the energies. This is the energy here expressed in kBt, and these few units you see in kBt, which morally is an hydrogen bond, strong hydrogen bond. But anyway, the point is that with no further assumptions, the stupid model predicts that you have this kind of mechanism, you can have this kind of mechanism in the weak biochemical range, which is where you would expect them to be, because usually this kind of architectural control are regulatory. You don't want to have permanent covalent bonds. And you see that the corresponding scale on the x-axis in concentration is fractions of micromoles per liter, which is the right order of magnitude of the order of magnitude of transcription factors concentrations in the nucleus. So in a sentence, with no big assumptions on the details of the model, roughly you see that this kind of mechanism, thermodynamic mechanism, can take place in the range of energies and concentration where roughly you would expect them to be. But there is something else I want to say about this phase diagram, because this is an oversimplification, but suppose there is something true. If we have such a switch mechanism, we can understand how conformational changes can be reliably regulated, sharply regulated. If you want to move, for instance, from the open to the compact state, just increase the concentration of binders. And as soon as you cross the threshold, you're 100% sure, with no fine-tuning, to have the new architecture, the compact one. And this is a standard strategy used by cell. Upregulate the genes which produce binders, and the trick is done. So with simple standard strategies, you control an architectural switch. And similarly, you can move in the other direction. This is changing the affinity of the binding sites along the polymer. And again, this is a standard strategy, which controls a switch in architecture. And so if this scenario is correct, we understand how functional, key architectural transformations can be sharply controlled and regulated. Now, how all this compares with the high C and fish data. This is on the left in my slide. I have the contact probability for the strings and binders model. And as you expect, as you know, the contact probability distribution depends on which phase you are in. If you are in the open regime, your counter probability is the blue one, and your counter probability is a power law with an exponent, which in this model is 2.1, because this is the self-avoiding walk exponent. Now, when you move instead to the compact regime, the counter probability develops a plateau, you see. So if you want to fit there with the power law, you can, but the exponent is 0. And in fact, this is how the exponent behaves as a function of the concentration of molecules, for instance. You see, you have this sigmoid behavior. At low concentration, you have 2.1. At high concentration, it's roughly 0. And in the narrow transition region, you have a range of values which connect the two plateaus, the two extremes. Now, I think importantly, if you want to compare this with real chromatin, we have to keep in mind that chromatin is likely to be a mixture of states. So with this toy model, it's very easy to see what happens when you consider a mixture of differently folded states. So by now, let's forget about the theta point fractal state. I will show you later on that it is likely to play a role in chromatin. We have data to show this. But by now, let's keep things simple. So let's look just at this, in this model, at a mixture of open and compact polymers. And I call F the fraction, this is an abuse notation, the fraction of open polymers in the mixture. And you understand, but by changing F, you change the counter probability. If F is 100%, you have only open polymers, and so your exponent is 2.1. If F is 0, you have only compact polymers, and so the exponent is 0. And you see here how the counter probability behaves as a function of when you change the fraction of open polymers in the mixture. And you see these three values are chosen just to play around, and 60% of open chromatin gives the green data, which is this one. And you see, in about a decade, you can fit with a power law with an exponent which is, that fraction is chosen to get an exponent which is close to 1.08. And you see, understand the story. If you decrease the fraction of open chromatin, so you get more compact mixture, you get the yellow data, 45% of open chromatin, and you get an exponent which is roughly 0.93, which is close to what you get with chromosome X. And even the banding of the distribution is found. And similarly, chromosome 19, if you increase F to 8%, you get the bluish data, exponent roughly 1.3, close to chromosome 19. Now, what about the mean squared distance in this model? Again, two behaviors according to the phase you are, plus the theta point, which by now I'm not discussing. And in the open phase, you get the power law, known power law for the self-avoiding walk. This is the mean squared distance as a function of genomic distance, sorry. But crucially, in the compact regime, you get a plateau. And for that reason, you can fit also the kind of fish data I mentioned at the beginning. These are the two examples I showed, there's plenty of. And you see, this is the fit with the strings and binders model in the compact regime. The fit isn't good, the fit isn't perfect, but at least the trend is captured. And so if I have to summarize what I've said up to now, the impression is that with very simple tools from polymer physics, we can understand the variety of fish data, power laws, plateaus, and the range of exponents observed experimentally with, say, the high C methods. But there is something more I want to say, which is this. In polymer physics, we are well used to consider the so-called moment ratio of the distance between loci. This is loci is the notation from biologists, of sites, notation from physicists, which is written there. It's more complex to say than to read. But anyway, this is the average of the fourth power of the distance between two sites divided by the square of the average of the squared distance. You know, this is a very important quantity in polymer physics because this is dimensionless, and so it's very easy to compare with real data. And furthermore, it takes universal values in different models. So for instance, in the self-avoiding walk or in the ideal Gaussian chain, you get exactly 3 L's for that quantity, for moment ratio. And you see the behavior in the strings and binders model. If your concentration is small, you are in the open regime, or if it is high, so you are in the compact regime, you find roughly 1.5, as you would expect. But you see, around the theta point, the moment ratio is significantly different from 1.5. And so this is the thermometer which we can use to probe locally which is the state in which chromatin is. If you find something close to 1.5, if this scenario is true, you should be probing a compact or an open state. If you find something which is far from 1.5, then you can be at close to the theta point fractal state. And there is data on that. There is a brilliant paper by Mateusz Langer et al. In PNAS 2009, where they have sampled this in a range of different loci, chromosomes, types of cells, and genomic distances. And we have collected their data in this picture. And you see, to explain very briefly this picture, you see there is an accumulation point at about 1.5, which in the view, what I'm telling you is saying us that, well, a relevant fraction of chromatin is indeed in the compact or in the open state. But there are significant departures. And in the view, what I'm saying, this means that a fraction of chromatin is not just compact or open, but can be in a state close to the theta point state. And to my surprise, even the scale you measure, remember this is dimensionless, even the scale you measure experimentally is close to the one you have in the strings and binders model. So stupid model explains also the key quantity, which is the moment ratio. So the picture which is emerging out of this, putting together the data I mentioned, the stupid model I mentioned, is roughly this. Chromatin is a complex mixture of differently folded regions. With the high-C methods, usually you access averages across genomic loci, across different cells, and so on. So chromatin is likely instead, the data point to the direction that chromatin is a complex mixture of differently folded regions. And you may have a given locus which is in a compact state, another which is in an open and theta point fractal maybe. And maybe the mechanism, the thermodynamic mechanism, standard mechanism of polymer physics, which can compact and change the state of a locus, as I told you, could be responsible for the local organization. And of course, different loci have different binding sites and so on, different transcription factors which bind and so on. When you look at the average counter probability from IC data, you're blurring things. And so you get an exponent which is in a range around 1 because it is an average across different states, open, compact, and theta point fractal. So maybe I was too quick, but these are my conclusions. The strings and binders model illustrates in a very simple way how the architectural patterns can be established and regulated in a Brownian system of polymers interacting with binding molecules, Brownian binding molecules. And the idea is that the stable folding classes, the states, folding states you find are just they correspond just to the emergent thermodynamic phases in the system. Of course, side by side with that, you have a number of off-equilibrium states, including the fractal globule states we have listed before. And in this context, we also understand how conformational changes, which are vital in a number of cases, can be sharply and reliably controlled by simple strategies, such as upregulation of a gene or chemical modification of chromatin. And again, the impression with the current data is that the range of high-C and fish data that you can find can be simply, again, explained in this framework. And you can explain the mean squared distance, the high-C power, the counter probability exponents, the range of exponents, the moment ratio, and so on. And so the picture which emerged, again, is that chromatin is likely to be a very complex mixture of differently folded regions, which can be controlled by thermodynamic mechanisms, as those we know very well in polymer physics. Of course, whether this scenario is correct is still to be understood, and I thank you for your attention.
Thank you very much. That was, for me, very kind of bringing different. You explained those different scaling exponents. I had great problems understanding this, so for me that's a very good explanation. Yes, so
I like your model very much. However, there is one factor to consider, that is that these various DNAs coming from various chromosomes can have varying elastic properties, right? Different persistent lengths, non-uniformity, and stuff like that. And maybe this fudge factor F that you have is just related to the difference in this non-uniform elastic behavior, because that controls the way the polymer is moving.
Absolutely. This is precisely my point. Details matter. So you have a general scenario which is dictated by the standard views we have of thermodynamics of polymer physics. But of course, if you want to look at a specific locus, its details, persistence length, state of chromatin, chemical modifications, all this is going to matter.
yes, Aris?
So that was a great talk and really, really interesting ideas. There was one thing, though, that I kind of was expecting you to do throughout the talk, and then you didn't do it, and I was curious about it. So kind of the simplest version of, I feel like, the kind of model that you're describing, if we wanted to apply it to data, is, there's the open stuff, there's this compact stuff. Well, already in the high-C context, one talks about, this like open and condensed, thing, and the two compartments, et cetera, et cetera. And you point out that the open fraction determines what the slope ought to be, and that this varies from chromosome to chromosome, all of which is true. It suggests that because actually the open fraction varies enormously from chromosome to chromosome, that one ought to look using those, principal eigenvectors at what the open fraction is on different chromosomes, predict what the slope ought to be, and see whether there's a match, right? And obviously that would be the easiest way to very strong evidence, right? Or strong evidence that this is well supported by the data. I was kind of expecting you to do that, and I'm curious whether you've tried it and whether you've been thinking about that.
So you missed the first question I thought, because what I showed at the beginning is precisely that different systems have different exponents,
Yes.
different chromosomes, different exponents, and so our simple parameter f, which is a very I stress this. This is very schematic. For instance, in that mixture.
All I'm saying is that the parameter f, there's a very natural interpretation for human chromosomes, which is that it is the fraction.
Oh, no, I'm less favorable to my model than you are, because this is a schematic model, as we just said. It's not just the fraction of open, compact, and remember, theta point fractal states which matter. But it's also other local details which are going to define exactly what's your average exponent. This is only a toy model to show how this is in fact the case. And for instance, with the moment ratio data, you can clearly see that in fact those three states identified by this are likely to be found in reality.
That said, right, if you did try to use the open fraction as your f, and you saw that there was a pretty decent correspondence with the slopes of individual chromosomes, I would not call that bad news. I would think that that's
absolutely. This is our point. You can explain with very stupid model all those different data.
Yes, please.
I have a question. I don't know if I understand pretty well your model, but did you think about another mechanism in this chain that could be the distribution of the sticky points along the chain?
Absolutely. You are quite right. We think this is one of the sources of specificity.
Yeah.
In a number of cases, this is going to be important. At the level of, and we are working on that, we have data on specific locus where we have tracing the binding factors and the binding sites, and we are making experiments on that. But this is to come. But this is precisely the point. here the aim is only to show with a very simple system where the binding sites are equally spaced along the string, what you get.
Chris?
And just confirm that these are all equilibrium results that you're getting.
All equilibrium results. Of course, you can consider off equilibrium, and this opens a door.
Okay, if there are no more questions.